Hello, everyone, and welcome to today's webinar, Achieving Operational Excellence in Clinical Chemistry Through Innovative Assay Design and Technology. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott. To learn more, visit them at corelaboratory.abbott. Now, we encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Q&A, ask a question box, and click send. We'll answer as many questions as we have time for at the end of this presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. This webinar is educational and thus offers continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain those credits. I'd now like to welcome our speaker, Dr. Marvin Berman, a manager of scientific services at Abbott Core Diagnostics. Dr. Berman, welcome. You may now begin your presentation, sir.
Thank you. Just to give a little background about myself, I have 30 years of experience at Abbott Labs. I've held a number of positions that look at the product life cycle. I've got expertise in developing and supporting new products, as well as spec setting and product investigations. I have expertise in clinical chemistry and immunoassays, and I'm the contact for diabetes and metabolic syndrome disease. Learning objectives today. For decades, clinical chemistry has been a key discipline in lab medicine and comprises the majority of the workload in the core lab. In this one-hour webinar, participants will learn how recent innovations in assay design, analytical technologies, and instrumentation are still being made and are helping to deliver high-quality results and take laboratory operational excellence to the next level. Abbott has a great history of innovative design in diagnostics. Started with manual assays in the early 60s. The first immunoassay for HBV and HIV. We are well known for our blood screening assays. We have gone into automated instrument systems like molecular diagnostics, our immunoassay and ClinChem systems, and real-time PCR. And Alinity is our next generation of informational systems. As well, Abbott has a great history in clinical chemistry. Sorry about that. We have been developing ClinChem instruments from the 70s onward. We're well known for our Architect platform, and now we are placing our Alinity instruments for immunoassay and ClinChem. How does Abbott help the labs? We have more than 40 years of expertise in clinical chemistry, and we provide personalized solutions to give you the power to address the challenges of facing your lab. We have assays with great performance. We maximize throughput in a very compact footprint. We provide confidence through our thoughtful design, and we are always responding to changing needs of the laboratorium. What is Sigma Strong? Sigma Strong is Abbott's new approach to clinical chemistry, where we are bringing assay formulation improvements and continuing to leverage our innovative technologies to deliver confidence in Six Sigma clinical chemistry results, which I'll go over in the following slides, assay productivity through assay and system design, and leading to a total value of ownership improvements, enabling labs to achieve sustained operational excellence. I'll go over assay designs and impact on operational productivity. Quality is essential in the clinical chemistry labs. The challenge, labs need to provide reliable, high-quality results to support clinician decision-making. How can we address that challenge? Assay parameters align to the latest CLSI guidelines and delivering Six Sigma analytical quality. The challenge is to simplify the clinical chemistry evaluation process with transparent assay performance specifications. CLSI guidelines are protocols that are developed from input from manufacturers, clinical labs, regulatory agencies, and Abbott uses those guidelines to ensure that our products meet not only our manufacturing specs, but the needs of the clinical labs. These CLSI guidelines are harmonized so that whether you go to Abbott or another vendor, you can be assured of the performance meeting the clinical lab if you're following these guidelines. For example, sensitivity guideline EP 17A2, which is known as LOB, LOD, LQ, provides guidance on three metrics: limit of blank, or LOB, limit of detection, which measures LOB in the test replicate of a known sample with a low concentration of analyte, and LOQ, or limit of quantitation, which is the lowest concentration that can be detected based on a target value for both bias and precision. The highlights of this CLSI guideline is that it allows for defining LOQ by precision only or by total error. If you're using less than 4 reagent lots, you can calculate your sensitivity LOB, D, and Q by instrument and lot, and you can take the worst case. Testing usually is 3 reagent lots and 2 instruments to really get the performance of your product, and allows extreme standardized deviation tests to identify potential outliers. As I said, testing is 6 instrument reagent lot combinations over 3 days. You have a 0 analyte sample of 60 reps, and you have low analyte samples to provide 60 positive reps. Data analysis for LOB, or limit of blank, is the 95th percentile of the 0 analyte concentration. LOD, or limit of detection, is your limit of blank and the standard deviation of your positive results. LOQ, limit of quantitation, is the estimated total error, and it's also the higher of the limit of detection or lowest concentration in which the total error or percent total error is met, or the lowest concentration at which precision is met, or the lowest concentration at which bias and precision are met independently. Acceptable modifications: you can have fewer 0 or low analyte samples, but you must test more replicates. Another CLS guideline is EP34. If you look at the labeling of manufacturers, it's very difficult to understand the range. Some call it an assay range, some call it a calibration range, some call it a reportable range. There are multiple names, as you can see in this slide. EP34 is harmonizing that to give you a measuring interval and a reportable interval. And I'll go into more detail on that in the next slide. The analytical measuring interval, or AMI, is the range in which specimen concentrations are measured within the medical and lab needs for accuracy with no dilution, concentration, or other pretreatment that is not part of the standard or routine measurement process, inclusive of dilutions or pretreatments that need no user intervention. Extended measuring interval or EMI, is the range in which concentrations are measured with appropriate accuracy by diluting the specimen before taking a measurement with the developed measurement process. And the reportable interval, or RI, is the range that includes the analytical measuring interval and the extended measuring interval, but also extends to the lower limit of detection, LOD. So you can see in the figure below that the AMI goes from the lower limit of quantitation to the upper limit of quantitation. The extended measuring interval goes from the upper limit of quantitation to the upper limit of quantitation. And the reportable interval encompasses both the AMI and the EMI and this lower limit of detection. So CLSI allows harmonization across different vendors to provide a consistent and clear definition of assay performance characteristics. Six Sigma quality is an analytical quality test which is defined by accuracy, precision, and a quality specification such as total allowable error for that analyte. You can see the formula on the right here, that sigma equals total allowable error minus the bias divided by the precision CV or SD. You have a certain target for your analyte. The precision of the instrument to measure the analyte is the spread here. The bias is how far are you from the target. And from that, you can calculate the number of defects, or a sigma level. 1 sigma means you have as high as over almost 700,000 defects per million, which is deemed an acceptable performance. 3 sigma is 67,000 defects per million, which is considered marginal performance. And 6 sigma, where you want to be, is only 3.4 million defects per million, and is considered world-class. So knowing your sigma kind of gives you a better sense of the performance of your product. Why is that important? It's important because it evaluates your specification of total allowable error against your bias and precision performance. And this just shows that using this equation, you can calculate your bias as well. It's a predefined total allowable error, and your observed bias and CV, your sigma value can be determined. Why is that important? On the graph here, you have accuracy plotted against precision. If you have poor precision and poor accuracy, you're going to be well off your target. If you have poor precision but good accuracy, you'll be close to your target, but you're going to have a wide distribution. If you have poor accuracy but good precision, your values are going to be very tight, but you're going to be off target. And where you want to be is in this grid here, where you have good accuracy and good precision to have high confidence that you're delivering a good response. This slide here shows that your limits for total allowable error, in this case from CLIA, are always changing, and they're getting tighter. So the better sigma performance you have, the better off you're going to be able to meet total allowable errors in the future, as these limits are only going to get tighter. This is some quality data from Westgard, where he pulled QC data from participating around the world to assess different assays from different competitor systems. And you can see in this company here, there's a wide spread of performance of their products. It's better in this company where you have more products in the 6 sigma range in green, and less in the 2 sigma range. And here is where you want to be with a product that has a lot of their products in the 3 to 6 sigma range. Again, here's another example from Westgard, where he shows Abbott's Albumin assay against Atelica and Cobis for a wide variety of assays, and determining if they're in the range of 1 to 6 sigma. And you can see plotted here the number of 6 sigma assays for the different assays, and you want to have a platform that has a high number of assays in the 3 to 6 sigma range. This is an example of Abbott's 6 sigma strong assay for Albumin BCG. As you can see, it has good accuracy, 4.8% bias against a target of 3.72. It has good precision of 1.4% against a total allowable error goal of 14%. It has a percent total allowable error of 7.6 and a sigma greater than 6. And so it's in the 6 sigma range of world class. Now some of the other sigma strong assays performed. Here's again Albumin BCG, another albumin, BCP, amylase, cholesterol, and total protein. And you can see all of these assays are 6 sigma or greater. Why is it important to have a 6 sigma assay? You can see that the Westgard rules for higher sigma assays are better than for a low sigma assay. So over here you can see a 6 sigma assay, and here you can see a 3 sigma assay. 6 sigma assay quality requires only 1 control rule with 2 control levels, where an assay with less than 4 requires an 8x rule with 4 quality control measures in each of 2 runs. So would you rather run 2 control levels or multiple controls? What is the impact on Six Sigma metrics on a particular lab? Here's an example of a lab where they had a 91% reduction in false QC rejection after implementing Six Sigma quality, and 91% labor cost savings when they use assays with greater Sigma performance. Six Sigma quality goes beyond the lab. You get decreased errors resulting in consistent delivery of accurate test results, improved turnaround time, leading to better patient care, better physician and patient satisfaction. So all around satisfaction, not only for the lab, but for the patient as well. I'll talk a little bit about assay productivity through Abbott's assay and system design. The challenge of the lab, you have many manual processes that hamper efficiency, increase risk of error and inconsistency. How to address that challenge? Use assays and systems that utilize innovative technologies that enable streamlined workflows, reduce hidden costs, and improve efficiencies. Abbott offers unique and proven technologies. An ICT module which provides a simple to install integrated chip for measuring the electrolyte sodium, potassium, and chloride with CVs of less than 1%, and each module delivers multiple tests and automated maintenance is provided. Clot and bubble detection to ensure that samples don't have bubbles or form and clots, which could affect sample integrity or aspiration of the sample. Smartwash technology, which prevents clinically significant sample-to-sample carryover and eliminates the need for additional consumables. FlexRate technology extends the linear ranges of enzyme assays for better first-time results and fewer repeats. And sample interference indices for preanalytical measurements of hemolysis, icterus, and leukemia to reduce the risk of reporting incorrect results due to interferences. And I'll go into a little bit more detail on these in the next slides. The ICT module is an integrated chip. It saves costs due to reduced operator interventions and reduced sample volume requirements. As I mentioned, you can get 60,000 determinations per module, less than 1% CV, automated maintenance. You can report one or all electrolytes with each sampling of the sample. So you don't have to do separate testing for potassium, chloride, and sodium. You get all those metrics at the same time from one sample. This slide just shows some of the performance metrics. You can see that tube-type equivalency across lithium heparin, sodium heparin, lithium heparin with separator tubes and serum separator tubes performance is equivalent. As well, endogenous interference testing for icterus, hemolytic, and leukemic samples were all well. Hemolysis for potassium was a little bit high, but that's because red blood cells contain potassium, and any hemolysis will result in the release of internal potassium. Sample and reagent aspiration. Our systems offer liquid level sensing and bubble and clot detection. Pressure differential technology accurately distinguishes between a normal sample, a bubble, and a clot. Linear software notifies the operator while the instrument attempts to remove a clot and clear the probe, and it reduces the risk of reporting incorrect results. So the software evaluates every sample. This is a graph of a normal sample, and this is a graph of a sample that has a clot, and the system can detect this difference and reject and flag a sample that has a clot. Sample carryover. Abbott smartwash technology ensures minimum sample-to-sample carryover. Smartwash provides an additional wash process using detergents and acid and alkaline solutions, optimized and automatically performed by the system as needed for reagent probes, sample probes, and cuvettes to reduce interference from carryover. The configuration of the smartwash parameters is determined by studies that looked at potential carryover between specific assay combinations. Abbott has established that carryover specifications meet or exceed the bias or total allowable error requirements for each assay. These specifications follow internationally recognized criteria from CLIA, NCAB, the EDCA-DEP, and several others are shown here. Abbott chemistry systems also have an optimum sampling sequence feature, which automatically determines the sampling sequence so as to minimize the consecutive measurements of assays configured as smartwash pairs. This just shows how we do these studies. You have a sample with the analyte that could potentially cause carryover, followed by a low concentration sample. You look at the threshold of the carryover, and you can determine that does the carryover meet the specification that you set to ensure that no carryover occurs. In this case, the cutoff for carryover was 0.022, and you can see that Abbott's architectural linear systems provide carryover less than 0.1 part per million, and it shows some other systems as a comparison. What is the impact of the heavy smartwash technology? No clinically significant carryover, and ensures confidence in results that influence patient care. It means no need for pipetting tips or alclotting to avoid carryover contamination in samples. No need to run all immunoassays first to avoid carryover in the chemistry systems, and no need for carryover evasion protocols that could lead to inconsistent turnaround times. One unique technology that Abbott offers is called FlexRate. It's an Abbott technology that extends the readable range of selected clinical chemistry enzyme assays by using two absorbance read time windows. A normal read window shown on the right in green, and an area read window shown on the right in orange. FlexRate allows extending the linearity of clinical chemistry enzyme assays by, as I said, using these two absorbance read time windows. It allows you to maintain analytical performance. It's automated, so the operator doesn't need to be involved. It increases the lab efficiency by getting results in the first pass, and it's a cost savings as well as time saving for better cost effectiveness. So as you can see, in green is a normal response that is linear in the green read window, and so it reports a result. However, sometimes your enzyme substrate can be depleted, resulting in a flat response in the main read window. So rather than requiring you to rerun the sample, the system defaults to an linear read time, and where your response is linear, and it can calculate a result on that sample without needing to retest. Here's just showing performance of some of our enzyme assays, and you can see they all have good slopes, high upper limit of detection, good lower limit of detection, and you can see that they perform very well architect versus alinity platforms. HIL interferences, what is that? It's a automated method on our systems that allows you to measure H index, I index, and L index, H standing for hemolysis, I for icteric or bilirubin, and L for leukemia. As you can see, the detection of the different indices, hemolysis in red, has specific absorbance at 415, 540, and 570 that can be measured. Bilirubin in yellow can be detected at 400 and 540. And leukemia, which is a little bit hard to see here in this slide, is over a wide range. By using the index values, we can estimate the potential for a sample to interfere. These indices cannot be used for determining actual concentrations, but gives a good estimate of hemoglobin, bilirubin, and triglyceride. Abbott has established qualitative scores of 1 to 4, as well as quantitative scores for each of the indexes, H, I, and L, in conventional units and SI international units. So, for example, an H index score of 4 means that the sample has greater than 500 mg per deciliter of hemoglobin. An I index score of 2 indicates bilirubin concentration estimates of between 4 to 10 mg per deciliter. Using these scores in combination with the package insert tells you if a sample could potentially interfere in that particular assay. And Abbott provides these package insert indices in easy-to-read brochure for all of its products. Conclusion. There are many considerations for assay and system design: commutability, correlation across platforms, standardization of assays, expected values, and caliper or pediatric reference ranges. Customer and key expert engagement, early adopter data, customer data and support, scientific collaboration. Quality. Abbott uses modernized expectations, consistent IFUs driven by CLSI guidance and other agencies, Sigma levels and accuracy tied to external standards, critical menu items like HbA1c, drugs of abuse, therapeutic drug monitoring that differentiate performance and function, efficiencies, error-proof designs, always on, load on the fly, onboard materials with high-capacity reagents, CALs and controls, lab utilization, small footprint, stable configurations, lab personnel, simple-to-use instruments, and high load capacities. Abbott can deliver values through innovative technology and assay design. Here's a case study from St. Francis Hospital that has been using the Abbott products. They've noticed that 89% of the Abbott assays were performing at 5 sigma or better. They saw cross-platform commutability with an average correlation coefficient of 0.994 for 32 of the Clinical Chemistry assays and 0.995 for 13 of their immunoassays. Operational excellence was observed by a 29% increase in tests per square foot, a 76% reduction in manual maintenance time, and a 96% stat turnaround time goal achieved for 3 critical panel assays. As well, labs across the globe have achieved measurable healthcare performance with the Abbott ClinChem assays. Here's from Clemenceau Medical Center. Resource utilization, 33% fewer staff required during primary shifts for routine operation, 75% less overtime expense. They've seen operational excellence, by eliminating manual sample prep for tests requiring pretreatment, and a saving of 15.5 weeks of time, and a cost savings of 40% decrease due to less quality control consumption. Thank you for allowing me to present to you, and if there's any questions, I can take them now. Thank you.
And thank you, Dr. Berman, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Now, it looks like we already have some great questions coming in. Dr. Berman, our first question, will using HIL on the Alinity system slow down the throughput of testing?
No, not really. There are publications already out there showing that using automated systems, whether it's from Abbott or somebody else, do not take a lot of time and hardly impact your throughput at all, but are very important as a pre-analytical check of the sample to ensure that you're not getting interference from hemolysis, leukemia, or enteric samples. Thank you.
Thank you so much. And how would someone determine the precision and bias of the assay on a system to determine the Sigma-Matrix?
There's no one way to do it. You know, one way is not more preferential than the other. What is important is to get a good estimate, whether that's running multiple instruments or multiple days or multiple samples to get an estimate of your precision or accuracy. We recommend using an external standard like a NIST standard to measure accuracy. That way it's commutable across different competitor systems, and we recommend at least a 5-day precision study to get a good estimate precision across at least 1 or 2 instruments. Thank you.
And thank you. And I want to thank our audience for these great questions coming in. Dr. Berman, our next audience member says, why would one want to use a standard instead of calibrator material to assess accuracy of a method?
There's nothing wrong with using a calibrator material if you're on the same system. But because you're calibrating with that material, you may be a little bit, you may not get as accurate a picture of your accuracy as you would like. By using an external standard that has been certified as truth or an exact reference material, you can get a measure of that material not only on your system but on other systems, as well as knowing it's independent of your calibration and is giving a true measure of accuracy on your system. Thank you.
Thank you so much. And we have one more question coming in. Dr. Berman, why does competitor labeling sometimes have better limit of detection sensitivity than Abbott products?
That's a good question. If they're following CLSI guidelines for how you determine limit of blank, limit of detection, and limit of quantitation, the comparison should be very simple to do. However, a lot of times the labeling will say limit of detection, but if you look how they're actually determining limit of detection, they were actually doing a limit of blank. So you want to use a manufacturer that is following the latest CLSI guidelines so that you can compare the results that you're seeing on one platform against another platform. And using Abbott's products, you can be assured that they are following the latest CLSI guidelines. Thank you.
Dr. Berman, thank you for your presentation today. Would you like to provide any closing remarks before we go?
I hope I've given you a good sense of what Sigma-Metrics are and why they're useful to understand how your product is performing against different competitor systems so that you know you're picking the best product available. As well, if there's additional questions that you have, please forward them to us, and our Abbott representative will be more than happy to address your questions or send them up to us. Thank you.
And thank you again, Dr. Berman, for your time today and for your important research. And I also want to thank LabRoots and our sponsor, Abbott, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.